brown tide is caused by a eukaryotic microorganism called Aureococcus anaphageferens, and it's a microscopic plant that lives in estuaries around Long Island.
Brown tide is one of many harmful algal blooms. Harmful algal blooms are a global problem. They're caused by many different types of algae. Some of those algae actually produce very potent toxins. That's not the case for Aureococcus.
So brown tide is a concern because it causes mass mortality of shellfish like oysters, which is a huge industry in the Long Island estuaries, and it also causes eelgrass and other seagrass beds to no longer be able to photosynthesize because Aureococcus blooms so densely that they block out sunlight. The reason why we're studying this organism is because it's affecting a multi-million-dollar industry and thousands of people who live right in our backyard.
Last year we had two very bad bouts of brown tide, and one in the spring, one in the fall, and that just nearly stopped the crop altogether. I would say I harvested maybe a third of what I thought I was going to bring in.
This study was really an exciting opportunity to follow on from an earlier study where we mapped the Aureococcus genome. It was the first harmful algal bloom species to have its genome sequenced, and having that genome sequence is almost like a decoder ring or a Rosetta Stone. It lets us look at the organism in a whole new way and really understand the very small scale, what we would call molecular level interactions or processes that are allowing that cell to work.
When you have just the organism and its genome, you're really boiling down to the essence of the organism. And then when you get to the gene expression level, you not only do you know what it has with regards to its genomic capacity, but you know what genes it's engaging to thrive under a particular set of environmental circumstances, and in fact the circumstances that we see in the bays here today.
Moving forward, one of the real challenges is to take what we've learned in the laboratory and be able to track those activities and those signals in the field. So we've mimicked conditions that we think are occurring in the field and learn important new information. But wouldn't it be powerful if we could go out and during the course of a bloom really track the activities and confirm what Aureococcus is doing?